Okay, so today we're going to cover the membrane, because that's where the action is, and we're going to even do a very basic review of membranes. But this is really just to fill in for those of you who want to review that, for those of you that want to learn about that, but really the main focus will be on ionic interactions, ionic distribution, okay, and what we call the resting potential. Now, don't let this scare you, but this is, I show this for several reasons. This is what we call an equivalent circuit for a nerve membrane, whether that's the axon or in the cell body, but particularly in the axon. And it first shows you that we can do such a thing. So we're talking about electrical signaling, so you can break it down into basic elements. And so I don't know if many of you are familiar with this, but these are basically batteries, resistors, and that's capacitance over there. And so these are the three basic things that later in the lecture I will make them equivalent to what we really need to know, which is the fact that between the outside and the inside we have a differential distribution of ions. And we are basically going to talk about electrochemistry, but the electro part generates a voltage, so it's a chemical-based voltage. But in the end, these things, which are really the ion gradients, they are batteries. Batteries have potential energy. This is a fundamental success, really, in evolution that goes actually way back to very early stages, that you exploit differential distribution of ions for signaling. And it really is remarkable how this works. But we need a couple lectures for you to really appreciate how remarkable it is. Now again, I know that most of you have seen a picture of an action potential. You probably learned already about a little bit about sodium and potassium from earlier courses, maybe even in the CMB course. But nonetheless, we are going to really delve into this. And when I was a senior in college, which was a million years ago, and even as a budding graduate student, you know, I thought I understood how this worked until you really are put to the test and realize, oh, you need to be able to make predictions. And when you are able to make predictions, you can really understand how the system works. So on Tuesday, I'm going to have a series of challenge questions. Today we'll have some questions that'll be a little bit more fun than not, I hope. They're to make some points. But these challenge questions will be about the action potential to get you really thinking. So if you understand the answers to these challenge questions, then you really will know how the membrane potential is formed, how it operates, and how the changes in what we call conductance or ion flow through channels produces the action potential. Okay. So, but in the end, we can use this equivalent circuit, and you see it's a cross. As I said before, all the action is in the membrane. Now, by the way, if you can't hear me in the back, let me know. If I'm standing in front of stuff too much, I can't do that. Sometimes I'll point with the computer, but let me know if you have any issues, all right? I'll try to be still. So it's the membrane, but you see there's resistance on the inside. Now, I think I said in the last lecture that nerves don't work like wires, okay? Well, they do have wire-like properties, so that any kind of voltage which is conveyed, and how is it conveyed? By currents. Currents can flow through the body of the cell or the axon, through the cylinder, but they don't operate like wires, right? They're not conducting electrons. Electrons are flowing. They're always left out of this picture. Always see sodium and potassium. Electrons, believe me, are there. You have to complete full circuits, but we ignore that. But they're not wires. But that doesn't mean that the relative resistance inside the axon isn't going to have an impact in terms of these voltage changes. So those are a couple of key things to keep in mind. We're going to come back to them. I want to just start from the beginning, and maybe I'll flick the light off for just a second. So you can see these cells. I alluded to the fact that we can recognize particular types of cells. And remember, we talked about this type of cell, this pyramidal cell. It's a lumpy pyramid. The cell body's in there somewhere, okay? This is color-coded, where you can see the dendrites, and as soon as the axon comes off, you get these collaterals very often. So all through the brain, and actually in particular layers in the outer portion of the brain called the cortex, which is all convoluted, right? in particular layers, we see these, and so they're recognizable right off the bat, and even in cortical elements in deeper tissues like the hippocampus. So we can say, look at that, and we can know that this is probably a processing system because by and large, as I mentioned before, this is the site where the major output happens from that part of the brain, from that little piece of brain. It's going to communicate elsewhere. So there's a lot of integration that happens, and then it goes through this. So we can see pyramidal cells everywhere, but if you look at any given pyramidal cell, they're a little different. And Carl spent his career drawing all these little differences, but in the end really didn't know what that meant. Now we do know that certain differences relate to function, and then we can overlay this on, as I said before, the types of ion channels that are here, but also the nature of the conductivity. But if you now look at other types of cells, you really do see the range. And the top is showing a series of different types of cells, which we learn a lot about in section 3 from the retina. So retina bipolar cells, so very simply with a cell body, you really have one dendrite at the top. That dendrite often has a few little nubbins, but it's really pretty simple, and then a basic axon that terminates. This cell has no action potentials, so it just conveys a voltage change. We'll come back to that. Now we find one that's called a retinal ganglia cell, and it has a dendritic tree that is quite expanded, okay, but a similar type of single axon coming down. So these look kind of like conduits, right? Like sort of direct line communication in terms of the bipolar cell. And you think even this one, but we know in the retina is that these dendrites spread out so that if light were coming through to influence this ganglion cell, which sits in the middle of the retina in a particular position, light surrounding that will influence its function. So just by looking at this structure, we would say this cell is collecting information laterally as well as centrally. This one, not. It's basically a direct line. And you can start making conclusions like that based on your understanding of the physiology. Okay? And then we get to something called an amacrine cell, which we're going to just mention in passing when we talk about the retina in the third section of the course. It has this quite elaborate dendritic tree, goes down to a cell body, and there's no axon here. It's just all dendrites all day. Okay. This cell generates an action potential. So this is a very weird cell. That's all you're going to really learn about amacrine cells, even when we get to section 3. Now we get over here to, these are specialized neurons, and it says mesencephalic. Don't be taken aback by some of this terminology. That just means the midbrain. But nonetheless, it's deeper in the brain, and just above what we call the brain stem. And we see that there's these cells, they have no dendrites, or very few. I'm not really sure what's going on there. I never really understood that. But by and large, they're a cell body and an axon. So now you're starting to see the range of it, and the ultimate, that Cahal thought was the most beautiful thing ever, was the dendritic arbor of what's known as the cerebellar Purkinje cells. You may recall, if you don't, the cerebellum's in the back of your brain. You all know about that, right? And that has a big role in coordination, although there's actually learning that goes on in the cerebellum. So these are, in terms of the integration in the cortex of the cerebellum, these are the big integrators that send their information down to some other cells that then go out of the cerebellum. But, I mean, this is a huge mass of branched dendrites. The other thing that's really interesting about these is this is really flat. It's not expansive in 3 dimensions. So there's like this planar, huge, almost circuit-like structure. But it's all focusing down on the cell body with an axon. This really is the ultimate in dendritic, what we call arborization or branching. Question, can you see it?
If they don't have dendrites, how do they receive information?
Well, remember synapses can be on the cell body.
Really?
Yeah. So yeah, okay, so I thought I mentioned this last time. The main contact, and so your thinking is correct. It said the main contact, by and large, is not the primary contact, and sometimes the exclusive contact of synapses coming in are the dendrites, but there are synapses on the cell bodies. So you're not seeing them here, but they're coming. So each of these cells has a function that has part of its roots in its structure. In some cases, it's really quite obvious, right? But in other cases, you don't know, because you don't know how fast that cell's conducting information. You don't know whether that ax potential looks like a regular ax potential. So in your textbook, you'll see this classic signature of the ax potential. They all look a little different. Okay? Some of them are wider, some are flatter, some have little bumps in them. But by and large, we're showing you the prototypic ax potential, but that might also be a difference here as well. Now, you're noticing, if you're looking actually at the PowerPoint, that the axon is colored a little differently. And this emphasizes a point that the nature of the membrane and the proteins that are, and the ion channels that are in the axon are different from the cell body and the dendrites. So not only is the cell polarized with regard to information flow and polarized in the nature of the structure that dictates that information flow, but the membrane and the proteins that are in the axon are different than out here, and there's basically like a gate here that controls even lipid movement, specialized lipid movement between those environments. So, the molecular underpinnings are what are dictating then how the signaling really goes. Okay. So let's just review what a membrane is. I think you will recall that it's a phospholipid bilayer. If you look at it in more detail, you see on the outside and inside, so this would be, there you go, this would be an expansion of this, that there's these phosphoryl head groups, okay? We say that they are polar, all right? The key thing is that they're charged. They're basically imparting a negative charge on the surface of the membrane. The membrane is very, very small. It's just some angstroms in cross-section. I guess you would call this the width or the height, whatever way you want to call it, some tens of angstroms, right? And so the surface on the outside and the inside, it's basically presenting a negative charge. And then you have the fatty acid groups on the inside, what they say is the nonpolar, but we really refer to this as hydrophobic on the inside, water hating, and hydrophilic on the outside. This makes sense. Water's interacting with this part of the cell, this part of the membrane, and water's obviously a lot more happy around a charged environment where that will allow it to interact and have its impact. Whereas inside here, you don't really find any water. Water does not cross membranes. Charged molecules don't cross membranes, pure membranes. Okay, they need to go through a channel, or you need a huge amount of it, okay, if it's charged. Or it has to have in that molecule a very large portion of the molecule, which is what we call hydrophobic. And that usually means it's a big ring structure. So for example, a lot of psychiatric drugs, they have big ring structures because that allows them to get in the brain because it's impermeable. You've probably all heard of the blood-brain barrier, right? Well, it's because everything's tight and the membranes don't allow things to pass. But even if something's charged, if it's really hydrophobic, sometimes, and there's enough of it, it'll worm its way through. So, but fundamentally, this is a basic barrier, right? I mean, this is the fundamental concept of a cell partitioning out, the outside from the in, so the inside can do different things than are happening on the outside. Now, this is just something in passing. Again, it's not something to scare you, but it turns out that in the brain there are a lot of specialized lipids. And a group of them are known as glycosphingolipids, but a particular subtype of those you'll find in the brain, particularly in axons. But they seem to have a similar function, is that they tend to organize in these what are sometimes called rafts. Now people don't like that term, they say microdomains. And they're in those, so here's the lipid bilayer, and then in those rafts, they're chock full of cholesterol. Okay? Cholesterol has a big impact on the membrane. They're not showing it here, but it actually widens the membrane too. So the membrane isn't just this uniform lipid bilayer. There are different types of lipids in there, and it's fairly clear now that these are sites that concentrate signaling molecules, and we now know as well ion channels. So on an axon or on a nerve cell or any other cell, it's not that the ion channels or the pumps or whatever receptors there, they're not just all uniform. You know, they're not just spread out. They really are often, if not always, localized. And sometimes they'll be anchored to the inside of the cell. So these are sites for receptoring complexes. So just to then complete the picture, what else are in the membranes? I've already mentioned most of these, not all of them though. So receptors, obviously a big part of communicating between cells, or just having the outside influence the inside of the cell, has to go through something. And so it was conjectured long before they were discovered that there had to be receptors for things. People gave drugs, you know, have an impact on behavior in an animal or in locomotion or something. And so, well, it's got to be interacting with something. For a long time I thought, well, drugs just go inside the cells and they do things in cells. Well, it's pretty clear, though, that most of the time, the vast majority of the time, it's interacting with a receptor. And we'll talk a lot about receptors. But just as a reminder, these are proteins that cross, or bridge, I should say, across the lipid bilayer. So that means they have a component of their structure that allows them to do that. And by basic prediction, again, virtually all the time, this part of that structure will be hydrophobic. And that's dictated by the fact that all the proteins, of course, are made up of strings of amino acids that form into secondary structures. But within, for example, these alpha helices, they tend to be the bridging part. So it's not shown here, but they're crossing the membrane. And they have, on the outside of the helices, they face out what we call hydrophobic amino acids. So that basically the protein is evolved so that it can sit and be just perfectly happy in this hydrophobic environment here, and then the rest of it will be hydrophobic. Okay. There are enzymes. Obviously, you usually think about an enzyme as being inside the cell, but they can be in the membrane. And fundamentally, for our discussion here, ion pumps are effectively enzymes. Okay? They just do something else. Most enzymes in the body, they're doing chemistry. Right? You have all this structure that makes this little pocket that allows basically chemistry to happen in that little confine, right? You know, break down glucose or whatever happens. Don't need to go into bloody details. But ion pumps, they're now actually doing something in a very active way. So skeletal elements, and then of course the ion channels, and then they're going to control both the resting and the action potential. So we're going to do that. Oh, I forgot to mention this down at the bottom. Okay, so sorry. Insulator and capacitor, what's that all about? So you have this membrane. It's by and large impermeant, certainly to charge things. It's hydrophobic, will go through, but by and large it's a barrier. But for purposes of our discussion, what's fundamental, particularly in the axon, okay, is that it's also a pretty effective insulator. All right, so it insulates against charge movement. Again, that fits with charged things not crossing. So sodium and potassium, they're just not going to go through this membrane. I mean, you have to put on a ton of it to the point where you break the membrane down. So sodium and potassium, all the ions, they don't go across the lipid. Okay, so they're strong, and we think about this in terms of electrochemical cell, it's a strong insulator, but it's also a fairly good capacitor. Now, why am I bringing that up? We're not going to really delve into this deeply in this course, but it's important to understand that charge, even though it's not going to cross, will align along the membrane, as it were. It will bind. So the membrane has a capacity to bind charge. And that has a fundamental effect in terms of charge transfer. Okay? So if you have ions that are flowing through channels, and a new ion comes on the inside of the cell, okay, some of it's soluble, but in terms of the voltage change, it has to charge up this capacitor. For those of you familiar with electricity, you know, electrical diagrams, that's probably quite familiar. But just to, in very simple terms, you will not get the voltage change that you want until you charge the capacitor. By and large, what it means is that it's starting to happen, and it just puts in a time element. It takes time to charge the membranes up, so what you'll see is a little bit of a lag. And so you see that actually at every synapse. When a neurotransmitter goes to the other side of the synaptic cleft, it opens up a channel or receptor that causes ions to flow, Okay, there's a little lag before then it will trigger off an action potential. Usually you don't see that in any of your drawing. You're going to see that. Okay, so the membrane is both an insulator for charge movement, but it also is a capacitor. So that's why in that equivalence diagram there was that Cm on the side that was for capacitance. Now why is that important? Well, certain cells have a lot more capacity, more capacitance, we should say. All right, they have more membrane. It would take more charge movement to charge that membrane to get a voltage effect. So there's an example in terms of signaling. If it takes longer to charge it up, that means that signal is not going to be as fast as a situation where the charge is more efficient. Okay, let's forge ahead. Now we'll talk first about ion pumps. Let me start with this before we talk about ion gradients. The real focus today, by the way, is going to be on understanding the ion gradients and what they do and how they contribute to the resting potential. On Tuesday, we're going to go back through the resting potential because you have to start there before you talk about action potentials. Okay? So this is a very basic, well, I use the word crude, but basic view of ion pump, the main ion pump, which is in all of our cells, okay, virtually all of our cells, okay, and it uses ATP. And it's an ATPase, meaning it cleaves ATP. So it's using the actual energy transfer of cleavage. It's using the high energy bond, the third bond in the ATP. It's using that to drive a conformational change in this ion pump. Okay? And it pumps sodium and potassium. Now, there are all kinds of pumps in the body. There's calcium pumps. In the kidney, there's a whole slew of pumps. Some of them are just transporters that are ATP independent. But nonetheless, they will drive the ion gradients. How are they doing that? Well, this is the outside. This is the inside. As you probably already know, the outside is rich in sodium, right? It's, our blood, and then outside of all of our nerve cells, all of our cells, it's rich in sodium. It's basically kind of like seawater, right? We use that connection. So rich in sodium outside, rich in potassium inside. So you need to maintain that, and that's what this does. So a lot of our time is spent keeping this pump happy, particularly in the brain, okay, whether you're thinking or not. We'll come to a little question later about that. So the sodium potassium ATPase is fundamental. If it doesn't work, then this won't work. So this creates and maintains the ion gradients, okay? And if you look at it in detail, this is actually a picture of it. It's very complicated, very interesting looking molecule. But in a more functional sense, we know that it opens up to the inside, okay? And this is a transition, all right? So this is before ATP is cleaved. Its default is it's opened up, okay? And sodium will bind to 3 sites, and potassium will be ejected if it's there, okay? But in terms of binding, any sodium that happens to be inside the cell, all right, it can bind into this pump, all right? And look what happens. ATP is cleaved. It drives a very dramatic conformational change, opens it up, but not only does it cause a conformational change, it changes the nature of these binding sites. So here, they're high affinity, meaning they have an affinity, they have a strong predilection to grab onto the sodium. You're all clear what affinity means, right? So they bind avidly the sodium with a conformation change. These sites now drop their ability to bind, so they release the sodium. And then the opposite happens with potassium. So now these sites become really avid or high affinity for potassium. So they load up, these go out, this loads up, and this will cycle back, okay? And it can cycle pretty quickly, certainly hundreds and hundreds of times a second, okay? So in this case then, it'll shift from high affinity to low affinity, potassium goes out, and it cycles and cycles. Now what do you notice about this?
Is it contributing to the concentration of positive charge outside of the cell? Because it's pushing out 3 positively charged.
Well, that was where I was going. So,
Oh.
so it's pushed. So you notice it's pushing 3 out, but only 2 are coming in, and they are equivalent valences. I mean, they are univalent, and there's nothing in terms of that valence that's different between sodium and potassium. I mean, they're different atoms, right? But or ions in this case. But, right, 3 are going out and 2 are coming in. So, what do you think that will do? Why isn't it 3 and 3 or 2 and 2? That's the first question, right? It's mismatched. So you're right, more sodium is going out equivalent for that
Well, it's
bit.
creating a buildup of positive charge.
It's building up positive charge. So that the question is it doing that? Yeah, but locally at the membrane, remember this is an insulator, all right? Even right there, right at that membrane, if you put one more charge out than you take in, that's a mismatch. What will happen? It isn't just, so overall, what is this doing? It's maintaining the high concentration of sodium outside and the high concentration of potassium inside. But they're floating around. But right at the moment that it's fluxing, that inequality right there, okay? What happens when you have an inequality of charge?
Molecules are more likely to move with the gradient. They just pass through
No, I'm
the
just
bottom.
talking about what's happening at the membrane, physically. What happens when you have, okay, we'll come to this later, but if you take charge and you separate that, what do we call that? Real simple. Starts with a V. That's voltage. This is what we call an electrogenic pump. This creates a small voltage right at the membrane. Okay. So in the end, in terms of the ion flow, that happens through the ion channels. They're elsewhere, so they're really seeing everything. So this is not coupled to those molecules. So that's an interesting question, but it doesn't work that way. So sodium goes out, gets dumped out into the pool. Potassium gets dumped out into the internal pool, and then from that pool, that's where you get the ion flow. But this is actually electrogenic. So if you inhibit this pump temporarily, you see a little bit of a voltage. And in fact, there is a cardiac drug that does this. I'm not going to tell you what it is. You don't need to know any more details than I'm doing. So this, again, creates and maintains the ion flux. And there are other things that will help to make sure that the total sodium and potassium levels are maintained outside. One of which is the fact that the balance outside is balanced by your kidney, right? And then inside there are other elements that can make sure that potassium is healthy. But by and large, this is how the gradients are formed, okay? So here's a nice little picture of the gradients, all nicely color-coded, all right? And right there, part of the deal is when you separate and have an imbalance of charge, okay? But fundamentally, just in terms of physical elements, if you take two charges, all right, opposite charges, and you do that, you will make a voltage. And if you do that, you get more voltage. All right. So if we just go through this, we see differential distribution, all right? So we have sodium coded here in blue. So the other thing that's being shown here is obviously when you're putting more outside, there's less inside, so that's the ion gradient, okay? Now we talk about equilibrium, and we're going to talk about equilibrium potentials in a minute, which is going to be the one thing that may be the most confusing bit today. But I think it's pretty straightforward that this is steady state, right? If this is an active membrane and ions are flowing, the sodium potassium ATPs is fixing that. So sodium and potassium are fluxing, not across the membrane like this, it's just showing the direction of the gradient, but through those pathways, right? But fundamentally, a lot of sodium outside, just a little bit inside. So you have what we call an inward directed ion gradient for sodium. The opposite is true for potassium, okay? Lots of potassium inside, only a little on the outside. The gradient for sodium is actually bigger if you compare a little bit, if you compare the difference in terms of the amount of sodium outside in compared to the potassium side. Now, there's somebody else here who we are going to ignore for our discussion for a little while, okay? And that's chloride. It's the green one. So chloride is inward directed. The only reason I bring this up now is chloride actually has an impact on the voltage if there is a change in chloride movement. But most of the time, certainly in the resting potential and with even the action potential, chloride just kind of moves around and doesn't really do anything. Okay? But it does serve an important purpose. And this is one aspect that can be confusing here. I'll bring it up now. We'll go on to talk about how the resting potentials form, but I'm going to bring it up again. The fact is that if you do the math, okay, and you add up all the charges on either side, they are electroneutral. So we have ionic neutrality. And this is something that confused me to start, but you have to go back to the original premise, right? If you have an unequal amount of charges out here, what's that going to create? Voltage, right? And that takes a lot of energy to be able to do that. So basically, when you add up all the pluses and minuses inside and out, they should come to zero. Basically all the time. All right? So what does that mean then? Why then is there ever a potential across this membrane? Okay. I'm going to go past this one pretty quickly. So these are the numbers where you can see this is a millimolar of the ground outside versus in. And then the chloride is the opposite of potassium. The other thing I want to say about chloride being the opposite of potassium, okay, so we have potassium so that positive gradient for potassium is facing outward. The negative one for chloride is facing inward. I'm going to tell you that the effects are basically very similar. They're certainly qualitatively similar in the same direction. Okay, so the voltage change when potassium goes out versus chloride going in is in the same direction. So there is this big gradient for chloride, and it can have an impact, but usually it doesn't for our discussion. Later in the course, we talk about certain neurotransmitters, particularly inhibitory neurotransmitters. They exploit chloride. Chloride moves, and it has a very strong pressing effect on the membrane. Not that the membrane, is pressed, but it's electricity. Okay, so here's where I want to start talking about how this works. And I think you've seen this before, and maybe you're really familiar. You may think, okay, I know this, great. Okay, but fundamentally, you got to start with first principles. And that is, again, the membrane is a strong insulator. It's an impermeable barrier. If there were no channels there, even though you have an unequal distribution of things, all right, and so here you're seeing the math, right? You have a lot of potassium outside and a counter ion to keep electron neutrality, all right? And this is inside, this is outside. So what we've done is we've done this, right? Outside, okay? So lots of potassium rich inside, very little outside. That's the gradient. No pathway for it, no permeability, no voltage. Nothing will happen. Oh, just to be clear, it's going to happen over and over again, but I just want to set the rule straight. We talk about a potential or a membrane potential. In that particular, talk about a membrane potential. We're talking about a voltage. Those will be interchangeable. In electrochemical cells, we talk about a potential, but we can measure it as a voltage. They are equivalent. The potential is often termed E, but it's usually equivalent to V for voltage, all right? So now, we put a little hole there that selectively allows the potassium to move. Okay. What does it want to do? Well, we don't know. We can't ask it what it wants. And there's no true force here. This is also a mistaken idea. There is a gradient that has an impact, but we're really talking about entropy and other elements that are involved here if you want to get nitpicky about this. So it's not a true force, but it's okay to say that this potassium being rich over here would rather somehow be over here. But on the average, what that means is that it would flow, the ion at the top of the gradient where it's most rich will flow to where it is less. Okay? There's physical forces that make that happen. This isn't a true force, but we can model as a force, and we'll do that on this. So you put a couple of holes in there, and they allow potassium to flow, but not really fast. Okay? Why do you want that? You have millimolar amounts, so you have big inside of cell, outside of cell, or big cells in your lab, your electrochemical cells that you set up with this membrane in the middle with the potassium channel. Okay. So there are lots of them here. There isn't just one big fat potassium, right? You got a lot of them, right? So if you put in a lot of holes and they conduct the potassium very quickly, okay, you will get a voltage, but what will happen? It'll just go away. You're going to collapse. We call it collapsing the gradient. That can happen in cells in pathology, right? What happens if you stabbed a cell, right, really badly, had a gaping wound in a cell, a lot of potassium would flow out, the gradients would collapse, right? So that can happen pathologically. So But you have a little hole, just a few of them. So it's selective for potassium, okay, flows fairly slowly. A little bit over time, it will come to a steady state equilibrium, right? So in a real cell, you know, the ATPase, and you may have other ways that the ions are going. But even if you ignore all that, a little bit of this is going to go over here, and it can't take that anionic charge with it. What have we done? What have we? We've separated charge, right? You separate charge, you get voltage. But the charge separation isn't like this. We're talking about right across the membrane, okay? Because all the other ions, they're floating around. They're not contributing. It's only right at the membrane. So basically, you see a charge distribution across that teeny little membrane, okay? And because those charges interact with the membrane, that has to charge up or charge the capacitor, right? Capacitor charges, this reaches a steady state equilibrium, then you will get a potential in electrochemical terms, all right? So the picture then is this. So now you have, on the balance, more positive charges just on this side of the membrane, relatively speaking, compared to this one. Now, what's that anion? Is it chloride? Well, if you do this right, you could just bank on the chloride. It turns out that in cells are full of proteins. On the average, the balance sheet of charge on the proteins is negative. So that's a big sink of negative charge. The chloride, you know, can move, so it's a little bit of a problem. But the channel for that is not usually open, but there could be transfers, whatever. So it's by and large, the inside of the cell is this negatively charged element, and you put just a little bit more potassium out here, just a little bit. Very little has to go. And now when you separate charge, you make a voltage. That's a lot of energy, isn't it? Right? We know that even at these distances, that's a lot of energy. Now, it ends up being tens of millivolts, but that's enough when you do that to have an impact on the flow of the ions. We call that the equilibrium potential, okay? And so that is signified by this E. Oh, it's over here, it's easier for you to see. Again, big box, huh? So E, usually we have the subscript is the name of the ion. So you can determine this for any of the ions, and that number, that potential, which translates into a voltage, is true for each of the ions. That's for sodium, potassium, and even chloride. They each have their own equilibrium potential. That is, if you had a channel there and it opened and it moved, that would be how the voltage would go, okay, to a particular number. I know this looks a little scary. Don't worry about all this stuff. That's the valence, the Faraday temperature, and so forth. So the gas constant. So, but the key thing of this is pretty simple. Can you all read this? This is the concentration, or really it's the ratio of the concentrations, but we don't care whether it's concentrations, whatever it is. It's a ratio of whatever there is outside, in this case potassium, over what's on the inside. So that voltage that you get depends on the gradient. The bigger the gradient, bigger the voltage, right? Now you're going to say, wait a minute, potassium is really rich inside. We have a problem, don't we? I mean, if I just told you that you have the outside over the inside, how does the gradient run for potassium? Inside to out, right? So what does that mean when you solve this equation? This is going to be a fraction, right? What happens when you solve this equation? It's a negative number. That's why you've seen already, probably, that a resting potential inside the cell when recorded is a negative number. It's like negative 60 millivolts because we have this. So this equation fits for all the ions. This is just the fundamental one for potassium, and it was devised by Neuroscience. Who was really quite a great scientist in terms of electrochemistry. And this equation holds true. But we can extrapolate for that. If we put sodium in here, we would get a potential, but there's a lot more sodium outside than inside. That's a positive number. Okay. Math always works, right? Now, I put a primer on Lalinga about the Nernst and something called the Goldman equation. The Goldman is an abbreviation of the Goldman-Hodgkin-Katz equation. Don't worry, you can read it, look at it. We might refer to it later. Certainly don't want you to memorize the Goldman equation. That not only puts in the Nernst equation in terms of the ion gradients, but it also inserts the relative conductance, the conductive ability, we call it permeability of each ion channel. So it makes it a more real picture, but it's a real big equation. So just want you to understand that conceptually. But it just goes step by step through this Nernst equation, and it solves for this to show you, if you know what's outside with potassium and what's inside with potassium, just from those numbers, you can calculate what the membrane potential should be, and it takes you through that. It's another way to read it. So fundamentally, this is important, though, just conceptually to know, and in terms of the basics, that is that you're relating the potential, the voltage that you get, to the gradient, and the gradient is the ratio of what's outside to inside. That's the fundamental concept here. Okay? Now, what's this down here? So when the voltage, okay, let's start here. We start here, we pop in some membrane channels, put in potassium channels. Ions start to flow. All right, and then you can put electrodes here and you can measure the voltage. So the voltage changes, it's going to go down toward -60 to -75, depending on your gradients. They can vary a little bit. They vary because of the amount of potassium inside the cell, right? That varies from cell to cell. So run that range, starting to go there, and then it stops. Okay, so why did the voltage stop? Well, we said, that's energy, right? You're separating a charge, and it takes a lot of energy to do that. So what is the ion gradient? It's a battery, right? It has this potential to do. So the potential has a limit, right? So that's the equilibrium potential, which we get from the Nernst equation. But when it goes down to that voltage, okay, it's at that equilibrium potential, right? That voltage that it creates then is basically going to lock the system. The ions, there will be no net ion flow, all right? So if you're sitting right at the equilibrium potential for any ion, but in this case for potassium, all right, it gets there, then there's basically no flow. If you tried to measure, not the voltage now, but the current flowing, it would go away. Initially, you pop the channels in there, and you get a nice current, you get a lot of current. Okay, so you'd see the current, and then it would just go off to zero. It would stop effectively. We measure current, though, as a net difference. That doesn't mean there could be a little bit of flux. Steady state, but fundamentally, operationally, it's very easy just to understand there is no net potassium movement. Why am I even telling you this? This is going to be important when we talk about the actual potential. Okay, now, I put these questions in, and I don't want you to come up with a number in grams here. Okay, I know this sounds almost crazy. So, talk about making these ion gradients. How much time do we spend doing this? How much energy do we put into it? Obviously, we're breaking down ATP cycles, right? But how much ATP is used per day to maintain an ion gradient? So I guess what I'd like to know is, do you think it's a lot? How would you approach figuring out how much is used? And the last part of this is this an important question? I mean, do we really care? They're working. We're not dead. You know, it's all, you know, things are buzzing around up there, so is this important? So let me ask first, how would you approach determining this? How much ATP is used per day to maintain our ion gradients? Now, we have ion gradients all over the place, okay? Not just in the brain. Any clue? When you're eating, what's the main point of eating? Ultimately, to make ATP, right? So what's the minimum amount of energy you need for the day? Often that's considered recommended minimum. Some people are
2,000
around there. 2,000, yeah. They say 1,600. Nutritionists say 1,600. I know people say, yeah, I do. I eat 1,600 calories a day for breakfast. So, and they're actually kilocalories. So yeah, that number, and I tell you that, and you can look this up from your previous biology text, that you use about 8 kilocalories to burn 1 ATP, right? So there you go. 1 mole of ATP. So that would mean you're going to turn over about 200 moles of ATP. That's just to keep the body going. But you know, we move around, we do a lot of things. So it turns out that maybe about 20 to 30% of all that energy that you produce from eating your food to make all that ATP, about 20 to 30% is used to maintain our gradients. So a lot of what you're doing every day when you're seeking out food is to make sure your gradients are happy. Okay. Now when you burn ATP, it also makes heat, right? And so the other thing is that you have heat in your body. All right. So more important question. So this is important to know because if we have an understanding in terms of how ion gradients are maintained in the brain, it can tell us something about brain metabolism. And what's interesting about the brain, you probably already know this, but fundamentally, there's no stored energy in the brain. There's no fat in the brain. There are fat-like things. There's these lipids, but there's no fat storage in the brain. There's no use of fat in the brain. It depends basically almost exclusively on glucose that's coming in through your carotids that goes into your brain, and a little bit on ketone bodies, but you don't use fat in the brain, so you're dependent on that glucose. So the glucose level, and there's some feeling that glucose changes, whether they're hypo or hyperglycemia, can have an impact on the system. Anyway, enough of that. So more importantly, if the ATP was rapidly depleted from a nerve cell, and you can do that using oxidative phosphorylation poisons, it'll go away pretty fast. How long will the ion gradients last? Well, again, I don't want a number here. You think they'll disappear right away? You think they'll last for a while? You think they'll last for a long time? Well, again, this emphasizes what ion gradients are. Remember our equivalence diagram? What are they? They're batteries, right? So again, away from the membrane, there's a lot of ions. So I'm here to tell you that when you have an axe potential or some kind of ion flux, it's usually very, very small, like 0.001% for each axe potential. So you can kill all the ATP, and if you take a nerve cell out and you run it, it will still generate axe potentials. Eventually they get smaller and smaller and smaller and they go away. But you might be able to do 1,000 or several thousand. So the ion gradients, point of this is the ion gradients impart this potential, but it's a big reservoir. So in the end, the ATPA is maintaining those gradients, right? But once they get the gradients in place, there's a certain stability to that, right? Granted, you don't really want your axe potentials to get smaller. You like to have them nice and big and uniform, because that's a signal. But nonetheless, that is the energy, that is the potential, okay? And so then how do they make signaling rapid and efficient? This gets to the next phase. Talked about it already. Well, again, the actions in the membrane, right? There is wire-like behavior of your axons, but they don't convey signals like wires. Okay. So now you have these ion gradients, and you want a rapid signal. How do they make that work? Well, it's the other side of the equation, which are ion channels, right? So the beauty of ion channels is you've got this potential ready to go, right? You got the, you've got the whatever way you want, whatever analogy you want, holding a brick out of a window, whatever. It's ready to go, okay? And all you have to do is you have a gate, and you open it up, and ions flow, and then you close it, and they stop. And these are molecular switches, so they can operate very, very fast. So the ion gradients coupled with the ion channels are why we can use electrical signals to be conveyed throughout the body through our nervous system in a rapid and efficient manner. So this is just a quick overview of ion channels and more of an introduction. Okay, so, and I'm showing two basic flavors of them that matter. There are others. There's actually water channels in certain cells. We're not going to worry about that. So for our discussion here, we really care about the channels that conduct ions. Okay, so in one category, the main category we're really going to focus on are the voltage-gated ion channels. Okay, so these channels, they're sensing voltage differences or really more voltage changes. In fact, the rate of change can be as important as the absolute change. And the reason for that, we'll see in a minute, is that these things are, like everything, a little more complicated than you'd expect. You think, okay, voltage comes along, they open, and then voltage goes away, they close. No, it doesn't quite work exactly that way. Okay. Overall, yes, but there's a little hitch in there. So they're made up invariably of multiple subunits, which we saw before already. The picture we had before was an ion channel. So these are individual proteins that traverse the membrane, okay? But they assemble in this lumpy donut, or cruller, or whatever you want. This hole in the middle, clearly that's the pore. That's where the ions flow, okay? All of the channels have multiple subunits. And the subunits can differ. So even though I've listed here a sodium channel, all right? Sodium channel family's fairly uniform, but there are different types of sodium channels. There's even variants. So they can behave a little differently. Now we come to, we're going to skip B for now. We come to the potassium, because we're really only interested in sodium and potassium right now, right? Come to the potassium channels, all right, same deal, but to the nth degree. There are at least 16 classes. I'm not sure the exact number. Somewhere between 15 and 18 classes of potassium channels. Depends on species and phylum. All right. So it's a huge number of potassium channels. There's all kinds of variants of them. They have all kinds of behavior. Some of them are weird, where they actually only conduct potassium going in, called inward rectifying. Those are weird. All right. But they can operate under certain conditions. So there's this huge family of potassium channels. So now just with those two, the sodium and potassium channels, we plug back into what I said in the beginning, that the array of ion channels and the behavior is going to dictate the function of that particular cell. You get a little, without actually giving you the bloody details, you have a particular type of sodium or batches of sodium channels and batches of potassium channels out in the dendrites of a particular cell type. That's going to convey the voltage differently than a neighboring cell that's a different type of cell that has a different array of these channels. So it all boils down in terms of signaling in the nervous system as to who's there and how they operate. Now there are calcium channels. We talked about them on Tuesday. You know, they're basically absent from the axon. Some people thought that they're there, and you can see a little calcium flow. It doesn't contribute to act potential, so we're not going to do much more with that now. But they're obviously a big trigger for release of neurotransmitter. And then I talked about chloride channels. And again, potassium going out will give an equivalence if you do it ion for ion. That voltage change when this moves, current, will be equivalent to the same flux of chloride coming in, right? And it makes sense, right? You're making this more positive, right? And you're making this more negative. So when the positive goes out, this is more negative, and when the negative comes in, this is more positive. You can play this game all day. But they are equivalent because they're going up. So they're diametrically opposed valences, charges, right? But they're going in different directions. That makes them equivalent. Okay, and then down here, there's some interesting channels that are 2 for 1 in the first case. They are neurotransmitter receptors. So there's a signaling molecule that interacts with the surface, really basically, we know now, kind of in between these, actually the interfaces between the subunits and this structure. So the receptors, but the receptor, when it's bound with something, in this case it's glutamate, okay, that's the major excitatory neurotransmitter in the body, in the brain, it opens a channel. But notice a big difference. It's showing sodium and potassium flowing. Now, they can't go past each other, right? They never flow at the same time. So in an instant, some sodium will go in, and an instant later, some potassium. But fundamentally, they're what we call non-selective cation channels. Okay? Whereas this channel only conducts sodium. And it's intriguing because there's a size difference between potassium and sodium and, of course, calcium. And you would think in some cases that, I mean, the sodium is really small. You think it'd go through the potassium channel. It just doesn't. So these are highly selective. These are selective for cations, and often they actually also conduct calcium. So they're a little more sloppy. They won't conduct chloride. So their impact in terms of voltage is different. But the key thing is they're not responding to voltage. Okay, they're responding to the signal that the synapse. But they give a voltage change. So they can start the voltage change, and then these guys sense that, and then they take the signal from there. Okay. There's actually some potassium channels that have a ligand because calcium binds to and affects the conductance change or the opening of it. And then there's some others. Okay? So just a quick introduction then to ion channels, but through all what we're going to be talking on Tuesday, they're going to be working underneath. Okay. And in fact, this now adds in what we're talking about. So in the equivalent circuit, these resistors, these are the pathways for ion flow. Here it's supposed to be electrons. But the resistors, they're the channels, right? Okay? They should have errors through them because they're highly variable. And then again, these batteries here, those are our ion gradients. And then in parallel, we do have capacitance. But if we have flow through the system, okay, in one part of the membrane, okay, that charge flow, okay, doesn't just sit there. It can flow in both directions, forward and backwards, all right? And so if we have movement of charge this way, it will go through, again, the resistance of the axon itself. So if you had a voltage change, let's say you had a glutamate receptor, it caused a little bit of a voltage change, because remember, it's also a channel. And then it started to activate a sodium channel. Okay. So if sodium started to come in, because it wants to come in, remember that the gradient goes inside, it's going to start causing a voltage change, but that's current. That current will start to flow down the axon a little bit. So the inside resistance of the axon does count. Okay. Almost done. So what I've done, I'm not going to do this for every lecture. I will try to do something equivalent to this. Is I've hit the key concepts at the end for you to know. Okay. Now, they can be packed with some detail, but these are where you should start. You should come back to this and say, okay, yeah, sodium, potassium, ATPase, that maintains the gradients. It breaks down ATP. You start, mentally adding into the detail, but fundamentally, gradients are formed because of ATPase. Each gradient, once it's formed, okay, has its own equilibrium potential. That is, its potential voltage that it could create if the ions flow. Now again, if you punch a big hole and that gradient collapses, all right, then you get nothing. So we're always talking about just a little bit of flow right near the membrane, a little bit of charge separation, and that charge separation is maintained, okay, that creates the equilibrium potential. All right? Now, we're going to talk a little bit more about this in detail on Tuesday, but the fundamental thing that we can say that largely accounts, but not exclusively, it turns out, because nothing's simple, but by and large, the membrane potential, the resting membrane potential, is due to what we call a resting or small conductance for potassium. Okay, so there's a particular type of potassium channel sitting in the membrane, okay, and it opens every once in a while. It opens a little bit, a little potassium comes through, and it does that until the voltage reaches the equilibrium potential, okay, and then it stops. Well, it turns out the resting potential isn't exactly at the equilibrium potential of potassium. We're going to find out about that. But it's the main culprit in creating that. And it's slow. All right, if you stop everything else and if you put the voltage in a funny position and you said, okay, let's let it go back, let potassium do its trick, it can take some time to get all the way back down to this. So it's sort of holding everything, but it's not fast. All right. So that equilibrium potential, that resting potential, it's fast enough, between signals, but it's not fast like a sodium channel causing an action potential. Okay? So, but that is the basis for the resting potential, and we can predict what that number is from the Nernst equation. If you know, you don't have to measure it. You could just know what's outside and what's inside. You need to measure those. You have an accurate number, and you have this resting, what we call conductance. Is everybody clear what I'm saying, conductance? I want to make sure, because everybody will think, yeah, I know what he means, and then later they'll go, no, I didn't. So it's what it sounds, though. It's conducting ions. So the relative ability or the relative amount of movement of ions will be the relative conductance. If we have a higher level of conductance, now microscopically that could be different. That could be more channels opening, but we don't care how it happens. If there's more of something in the membrane, in this case potassium channel, to conduct, so it has relatively high conductance, that means you're going to have a lot of ions flowing. Lower conductance, very few. So we know at rest there's only a few that are able to go. So but that just puts a time factor in how long it would get to the equilibrium potential. If you were able to open up a lot of those, it would get to the same position. All right, just get there faster. All right. So conductance is a relative term, but it really just talks about the ion channels and whether they're open. More of them opening, and some of them are inherently different. Some will conduct the potassium ions really fast, and some not so fast. And where is that happening? Inside that pore channel. Okay, the ions have to traverse through that pore, and in some channels they just zip right through. In other cases, they kind of take their time. So you could have inherently a difference in conducting the ions in the channel, but then it could also be that, you know, in this cell right now you have 10 of the channels open. Over here you have 100 of them. You have more open, more conductance. So it's a relative thing. All right? And then what we'll talk about Tuesday, the voltage-gauge ion channels. Obviously, they are the main signaling elements, right? So now you're starting to get voltage changes, and fundamentally we're talking about voltage changes away from the membrane potential. We call it the resting membrane potential because that means the cell's resting. It's not signaling. It's sitting there, just happy and, you know, waving around in space. But fundamentally, when we talk about these voltage-gated the ion channels, that means voltage has changed, the signal has happened, they respond to that, and then they provide the pathways for ions to flow across the membrane. Okay. So it turns out that this resting potassium conductance, this resting ion channel for potassium, has a particular name, I'm not going to tell you, but it turns out it doesn't really respond to voltage. It's just always got a little bit of a conductance, okay? So it's sitting there. So when all the other voltage-gated calcium channels have shut down and they're all quiet, then it takes over, and it's just, you know, like the little rabbit, you know, comes along and steady and sure, you know, it'll just bring that membrane potential back to rest, okay? So not all the channels are voltage-gated, it turns out, right? And this one isn't. Very, very subtle, but effectively no. It's the ones that are, they're the ones that matter for the action potential. Okay, we're done. Any questions? So we'll pick it up on Tuesday, and the plot will thicken when we talk about the action.